Human is a suitable environment for the growth of many types of microorganisms. Microbes are tiny living organisms found in water, soil, air, and other places, and they are small and found around us. These organisms are essential in our lives. For example, they can make us sick and play essential roles in human health. Type of microbes are bacteria, viruses, fungi, and protozoa. Human can get sick and many diseases such as food poisoning, septicemia, respiratory tract infections, and meningitis are caused by bacterial infections. Bacteria like Escherichia coli, Haemophilus, Pseudomonas, Staphylococcus, and Streptococcus can cause infections in humans. Microbes can be transmitted from person to person in many ways, including direct contact, inhalation of infected air, and consumption of contaminated food or water. Many microbes do not cause diseases. Many bacteria live on the skin surface without cause disease. Bacteria are single-cell organisms with a simple cell structure, and some bacteria have plasmid. The plasmid contain genes that give bacteria some properties, like resistance to antibiotics. Bacteria consist of a single cell with a protective wall. Some bacteria are aerobic, which require oxygen, and are therefore more likely to infect a surface area such as the skin or respiratory tract, while other bacteria are anaerobic, which do not require oxygen. Bacteria can cause symptoms of diseases by releasing toxin or provoking an inflammatory response. Each bacteria needs a specific environment to survive, like an environment with oxygen or without oxygen. Some bacteria need a heat environment while others need a cold environment. Examples of bacteria are Staphylococcus and Salmonella. They are good bacteria that play an important part in our health, such as lactic acid and probiotic. These bacteria live in the bowel that help us to digest food to make our body healthy. Other bacteria assist our immune system in fighting germs. One advantage of some bacteria is that it is needed to produce food such as yogurt and cheese. Infectious disease occur when bacteria invaded the human body. Some symptoms result from damage to human tissue caused by infections or toxin released by bacteria. Treating bacteria infection is necessary when the immune system have not overcome the infections. Antimicrobial drugs are agents used to cure without harm to the patient. There are two ways that antimicrobial drugs exert their effect on the target organism: by either direct killing of the organisms or inhibiting cell functions. These effects enable host defense to destroy microbes resistant to antimicrobial drugs. There are many classes of antimicrobial agents. Antimicrobial drugs divided into antibiotics, antivirals, antifungal, and antiprotozoal. And these drugs used to prevent and treat infection in human, animals, and plants. Bactericidal drug is one type of antibiotics. Bactericidal drugs are drugs that kill the organism against which they are effective. And these drugs do not kill organisms but inhibit the ability to replicate through inhibition of protein synthesis. Bacterial infections can be treated with antibiotics. Some drugs kill the infection's bacteria, whereas others prevent it from multiplying. Antibiotics are drugs that fight bacterial infections. Bacteria are very small germs that live inside human body, on the skin, and all around the human. Certain types of bacteria help maintain human health, while others can make humans sick, which is caused by mild to severe bacterial infections. These infections require antibiotics treatment, which help the sick person become healthy. Antibiotics as substance work by killing bacteria or stopping them from multiplying. The antibiotics kill bacteria by destroying critical parts they need to survive, such as DNA or cell walls. In addition, antibiotics prevent bacteria from making specific proteins to stop them from growing. Antibiotics can treat a wide range of conditions that affect many parts of the human body. Antibiotics can treat bacterial infections in different parts of human body. Examples of bacterial infections in throat and respiratory system are bacterial pneumonia, strep throat, and whooping cough. Examples of bacterial infections in urinary systems and reproductive systems are bacteria venijosis, sexual transmitted infections, and urinary tract infections. Examples of bacterial infections in eye are pink eye and orbital cellulitis. Examples of bacterial infections in other conditions are anthrax, enterochondritis, Lyme disease, and sepsis. Antibiotics have many benefits, such as successfully removing bacterial infections from the body, speeding up recovery, stopping the spread of bacterial infections to others, easing the symptoms of the diseases, protecting the patient from serious illness and complication, and saving lives. Some side effects of antibiotics include diarrhea, nausea, vomiting, dizziness, and rash. Examples of antibiotics are amoxicillin, doxycycline, ciprofloxacin, cephalosporin, clindamycin, metronidazole, azithromycin, and others. In vitro tests for most antibacterial drugs, such as the stability test, are based on the concentration of antibiotics required to inhibit growth. The test is determined by standard laboratory measurement of the mean inhibitory concentration. The MIC test divided bacteria into these susceptibility categories: resistance, sensitive, and indeterminate. And this test is used to determine if an agent will be effective or not against a specific organisms. Both antibiotic concentration and MIC are essential in determining drug doses. Bacteria are everywhere, in the air, on the mucous membranes of people's mouth and nose, skin, and intestine. However, the body immunological defense protects humans, and when pathogenic bacteria invade the body, a person will get infectious diseases. Bacteria can multiply, destroying tissue, releasing toxin, and threatening to spread via the bloodstream to vital organs such as the heart, brain, lungs, and kidney. The symptoms of infection disease depend on the sites of infections and type of bacteria. Identifying bacteria is essential to know which antibiotics to treat bacterial infections. The increasing use of antibiotics to treat bacterial infections makes certain bacteria resistant to antibiotics. These bacteria develop mechanisms of growth that are not affected by antibiotics, such as resistant strain of bacteria, methicillin-resistant Staphylococcus aureus, MRSA. Antibiotic resistance is a global health problem. In 1928, Alexander Fleming, who worked at St Mary Hospital in London, discovered the growth of Staphylococci bacteria had become contaminated with the mold of the genus Penicillium. He observed that bacteria growth in vicinity of the mold had been inhibited. Alexander Fleming isolated the mold, which produced an antibacterial substance called penicillin. Florey, Shane, and their colleagues at Oxford in 1940 analyzed the antibacterial effect of the substance, and they showed that it was non-toxic to the host but killed the pathogens in infected mice. Antibiotic drugs are usually safe and effective in treating bacterial disorder, ranging from minor infection to life-threatening diseases like pneumonia, meningitis, and septicemia. Since 1940, many different classes of antibiotics have been developed. Each class of antibiotics has a different chemical composition, which has antibacterial effects. Many antibiotics have a broad spectrum of activities against various bacteria. There are classes of antibacterial drugs, and within each classes there are drugs used against specific bacterial infections. Classification of antibacterial drugs are penicillin, cephalosporins, macrolides, tetracycline, quinolone, aminoglycosides, Lincosamide, Beta-lactam, Sulfonamide, and azole. Penicillin is known as an antibiotic obtained from penicillin mold. Penicillin G and V are two types of penicillin, and these antibiotics are effective against many infections caused by bacteria, such as Staphylococci and Streptococci. Penicillin is a member of Beta-lactam antibiotics, and Scottish scientist Alexander Fleming discovered penicillin in 1928. Benzypenicillin is a penicillin G compound, and penicillin was produced in 1942 by growing on the penicillin molds in deep tanks. The penicillin nucleus was produced in 1957, and semi-synthetic penicillin with different properties is synthesized by adding different side chain. Penicillin inhibits the enzymes involved in cross-linking the peptidoglycan layer of the cell wall, leading to osmotic repair. Some bacteria have beta-lactamase enzyme that hydrolyze the beta-lactam ring, which is bacteria's primary defense against penicillin. Bacteria also have a mechanism of modification of penicillin binding protein. This mechanism prevented from binding beta-lactam, reducing the permeability of the outer cell membrane of the gram-negative bacteria, quinolones. Some resistant bacteria may possess several mechanisms to act against penicillin. Gastric acid can easily destroy benzypenicillin. Penicillin is hazard with allergic reaction such as itchy, rash, fever, and anti-edema. Diarrhea is one of adverse effect of penicillin due to change in normal intestine flora, and this may process to Clostridium difficile associated diarrhea. Other adverse effect of high dose of penicillin is neutropenia, and rare adverse effect is anemia and thrombocytopenia. Benzypenicillin is active against Streptococcus pneumoniae and Streptococcus pyogenes. Combination of penicillin and gentamicin are active against Enterococcus faecalis. Over 90% of Staphylococcus aureus are resistant in hospital. Benzypenicillin drug is used for bacteria infections are Neisseria meningitis, Bacillus anthracis, Clostridium perfringens, Corynebacterium diffrae, Theribonemia baldum, Leptospira species, Actomyces australe, Borrelia burgdorferi. Some bacteria produce beta-lactamase, which open beta-lactam ring, and this terminate antibacterial activities of penicillin. Example of anti-Staphylococcus penicillin stable to Staphylococcus beta-lactamase are flucloxacillin, cloxacillin, methicillin, and oxacillin. Examples of broad-spectrum penicillin are amoxicillin, ampicillin, and co-amoxiclav. Amoxicillin is a structural analog of ampicillin, which is absorbed from the gut, and the oral dose of amoxicillin is 250 to 500 mg. Amoxicillin treats bacterial infections such as chest infection, pneumonia, and dental abscess. Ampicillin is a drug used to treat specific infection caused by bacteria such as meningitis. Ampicillin is acid stable, and the oral dose of ampicillin is 250 mg or 500 mg. Ampicillin is used to treat meningitis as infection of membranes surrounding the brain and spinal cord. In addition, it is used to treat infection in throat, sinus, lung, reproductive organ, urinary tract, and gastrointestinal tract. Co-amaxoclav is an antibiotic used to treat bacterial infections. This antibiotics contain amoxicillin mixed with clavulanic acid. Clavulanic acid has a beta-lactam molecules that has antibacterial activities and bind to beta-lactamase. The clavulanic acid prevents bacteria from breaking down amoxicillin, making the antibiotics work better. Co-amoxiclav is used to treat ear and sinus infection, throat or lung respiratory tract infections, skin and tissue infections, and joint and bone infections. Co-amaxoclav antibiotics is used to treat bacteria like Staphylococcus aureus, Escherichia coli, and Haemophilus influenza. Mesalinum is a beta-lactam spectrum penicillin antibiotic of the aminoglycoside class that bind to penicillin binding protein 2. Mesalinum is an oral drug that is active against gram-negative bacteria such as Enterobacteriaceae, and it is inactive against Pseudomonas aeruginosa. Mesalinum is mainly used to treat urinary tract infection caused by Escherichia coli. Penicillin is an antibiotic which is a semi-synthetic 6-alpha methoxy derivative of tetracycline, and it is highly stable to most beta-lactamase except metallocarbamidase (NOxa48). It is used primarily for the treatment of multiple drug-resistant gram-negative bacteria. It lacks activities against gram-positive and gram-negative non-fermenter such as Pseudomonas aeruginosa and Actinobacter species. It is active against some bacteria such as E. coli, Salmonella typhimurium, Haemophilus influenza, Klebsiella pneumonia, and other. Carboxyl penicillin is anti-pseudomonal penicillin, which has the same antibacterial spectrum as ampicillin. However, carboxyl penicillin can destroy Pseudomonas aeruginosa. Tetracycline is carboxyl penicillin, which is presented in combination with clavulanic acid to provide activities against beta-lactamase. It is used to treat gram-negative bacteria such as Pseudomonas aeruginosa and Protozoa species. Penicillin is anti-pseudomonal penicillin that is ampicillin derivative with a side chain derived from mure. Penicillin are used particularly against infections caused by gram-negative bacteria such as Pseudomonas aeruginosa. There are three riadopenicillin: azacilin, piperacillin, and meslocillin. Cephalosporin antibiotic were obtained from a filamentous fungus, Cephalosporin, in 1945. Cephalosporin impair bacterial cell wall synthesis that exhibit time-dependent bacterial killing. Examples of first-generation cephalosporins are cefazolin, cefadurine, cefaduroxil, and cefalexin. Examples of second-generation cephalosporins are cefoxistine and cefuroxime. Examples of third-generation cephalosporins are ceftataxime, ceftazidime, ceftriaxone, cefixime, cefprozil, and cefwodexime. Cephalosporin protect beta-lactam ring, which result in compound with improved activities against gram-negative organisms. Ceftobiprole is a barnetyl cephalosporin which bind to the mutated penicillin binding protein 2, responsible for methicillin resistance in staphylococci. Ceftolozane, tazobactam, and ceftazidime, avibactam, have extended activities against Pseudomonas aeruginosa. Macrolides antibiotics are bacteriostatic agents that block protein synthesis in bacteria by binding to the 5-TS subunit of the bacterial ribosomes. The macrolides antibiotic is used against gram-positive cocci, including streptococci and staphylococci, and some upper receptory gram-negative bacteria. Macrolides antibiotic are also used to treat vaginitis, otitis media, sinusitis, and bronchitis. The antibiotic drug is to treating Legionella, chlamydia, and mycoplasma infections. Examples of macrolides antibiotics are erythromycin, clarthamycin, and azithromycin. Erythromycin is a macrolide drug that bind to bacterial ribosomes and interfere with protein synthesis. Erythromycin, a broad-spectrum antibiotic, is effective against certain diseases, such as Legionnaires' disease, and it is a drug for microplasma pneumonia in children and diphtheria. Erythromycin is used against gram-positive organisms, and it has some activities against gram-negative bacteria such as Legionella species, Campylobacter species, and Bordetella proteaceae. Azithromycin is another macrolide drug that bind to bacterial ribosomes. It has activities against a number of gram-negative organisms, such as Haemophilus influenzae and Neisseria gonorrhea. Azithromycin is used to treat respiratory tract and soft tissue infections, sexually transmitted diseases, and cystic fibrosis. It is effective against traveler diarrhea. Clarithromycin is a macrolide antibiotics that bind to ribosomal receptor sites of susceptible organisms, inhibiting protein synthesis of the bacteria cell wall. Clarithromycin is a bacteriostatic that may be bactericidal with high dosage over very susceptible microorganisms. Clarithromycin is drug that act like erythromycins and has a similar spectrum of antibacterial activities. The drug is mainly against gram-positive organisms. Clarithromycin is used to treat bronchitis, skin infection, Mycobacterium avium complex, prophylaxis, peritonitis, pneumonia, maxillary sinusitis, and others. Examples of tetracycline antibiotic are tetracycline, doxycycline, tigecycline, dimiclocycline, minocycline, and others. Tetracycline is an antibiotic that inhibits bacteria protein synthesis by binding to ribosomes, and it is bacteriostatic. Tetracycline have activities against gram-positive and gram-negative bacteria. Tetracycline is used to treat inflammatory acne vulgaris, Lyme disease, Mycoplasma disease, Legionella infections, Rocky Mountain spotted fever, renal impairment, and cholera. Doxycycline is a tetracycline antibiotic that inhibits bacteria protein synthesis by binding to ribosomes, and it is used in the treatment of infection caused by bacteria and certain parasites. Deoxycycline is used to treat respiratory, skin and soft tissue infection, urinary tract infection, pelvic inflammatory disease, acute gonococcal infection, syphilis, and cholera. Periodontitis, malaria, rosca, and others. Tigacycline is a glycocycline antibiotic that is derivative of minocycline, which inhibit bacterial protein synthesis by binding to the 30S ribosomal subunit. And tigacycline is a bactistatic. Tigacycline is antibiotic used to treat a wide variety of bacterial infections, and it is used to treat community-acquired pneumonia, skin infections, and complicated intra-abdominal infections. Dimiclocycline is a broad-spectrum tetracycline antibiotics that inhibit bacterial protein synthesis by binding to ribosomal receptor sites. Dimiclocycline also inhibit antidieretic hormones, induce water reabsorption, and dimiclocycline is bactericidal. Dimiclocycline is used to treat a wide variety of bacterial infections, and it works by stopping the growth of bacteria. Dimiclocycline is used to treat acne, chronic itis, urinary tract infections, and complicated gonorrhea and Quinolone are types of antibiotics that inhibit the growth of bacteria. Nildicyclic acid was the first quinolone discovered, and it was effective against urinary tract infections. Quinolone antibiotic inhibit bacterial DNA gyrase topoisomerase, which prevent the supercoil of DNA. Quinolone are active against gram-negative organisms such as Pseudomonas aeruginosa, Mycobacter, and Legionella pneumophila. Examples of quinolone antibiotics are ciprofloxacin, dilafloxacin, gemifloxacin, levofloxacin, ofloxacin, and others. Levofloxacin is a fluoroquinolone antibiotic that inhibit the enzyme DNA gyrase in susceptible microorganisms, interfering with bacteria cells replication and repair. Levofloxacin is antibiotics that is used to treat variety of bacteria infections. Levofloxacin is used to treat bronchitis, pneumonia, acute bacteria sinusitis, urinary tract infection, black anthrax, colon ulcer, and others. Ofloxacin is a fluoroquinolone antibiotics that inhibit DNA grase in susceptible microorganism, interfering with bacterial cell replication and repair. And ofloxacin is used to treat bacterial infections. Ofloxacin is used to treat urinary tract infections, pelvic inflammatory disease (PID), lower respiratory tract, prostatitis, acute gonorrhea, bacterial conjunctivitis, corneal ulcer, acute otitis, hepatic impairment, and renal impairment. Aminoglycosides, antibiotic, are drugs that inhibit protein synthesis and it acts inside the cell by binding to the ribosomes. Aminoglycosides antibiotics are active against staphylococci and aerobic gram-negative organisms. Examples of aminoglycosides antibiotic are gentamicin, topramycin, amikacins, netelmicin, streptomycin, and others. Gentamicin is an aminoglycoside antibiotic that irreversibly bind to the protein of the bacteria ribosomes. Gentamicin interfere with protein synthesis of susceptible microorganisms and it is bactericidal. Gentamicin is used to treat acute pelvic, bone, joint, respiratory tract, burn wounded, skin infections, urinary tract infection, septicemia, meningitis, eye infections, and others. Topramycin sulfate is an aminoglycoside antibiotic that irreversibly binds to protein on bacterial ribosomes. Topramycin sulfate interferes with protein synthesis of susceptible microorganisms. Topramycin sulfate is used to treat skin, bone, joint, and respiratory tract infections, burn wound infections, urinary tract infections, septicemia, meningitis, eye infections, bronchopulmonary infections. Lincosamides are antibiotic drugs that prevent bacterial replications by interfering with the synthesis of protein. Examples of Lincosamide antibiotics are clindamycins, lincomycins, and others. Lincosamides antibiotics are used for infections caused by Staphylococcus bacteria, like methicillin-resistant Staphylococcus aureus (MRSA). It is used to treat bacterial infections in skin, soft tissue, mouth, respiratory tract, and gastrointestinal tract. Beta-lactam antibiotic are drugs used to treat bacterial infections. Examples of beta-lactam antibiotics are monobactam and carbabinium. Azithromycin is monobactam beta-lactam antibiotic that is active against gram-negative organisms, include Pseudomonas aeruginosa, Haemophilus influenzae, and Neisseria meningitis. Azithromycin is used to treat septicemia, urinary tract infection, and gonorrhea. Carbabinium are beta-lactam antibiotic used to treat bacterial infections. Carbabinium are drug used against gram-positive and gram-negative bacteria. Examples of carbabinium antibiotics are embiniums, merobidiums, dorobiniums, and artabiniums. Merobidium is a carbabinium that bind to penicillin binding proteins and inhibit bacterial cell wall synthesis. Merobidium is active against most gram-positive and gram-negative bacteria, including E. coli, Pseudomonas aeruginosa, and methicillin-sensitive Staphylococcus aureus, and merobidium produce bacterial cell death in most susceptible organisms. Merobidium is used to treat skin and intra-abdominal infections, meningitis, and concurrent bacteremia. Sulfonamides are antibiotic drug that inhibit nucleic acid synthesis, and it is a structural analog of B-aminobenzonic acid, PAPA. Sulfonamides compete with PAPA for the enzyme dihydrotyrate synthetitis, DHPS. Therefore, it inhibits the growth and multiplication of bacteria. Sulfonamides are used to treat bacterial infections, Crohn's disease, diabetes, and ulcerative colitis. Examples of sulfonamides are trimethoprim, sulfodiazine, and others. Azole are antimicrobial drugs used to treat bacterial infections. Azole antibiotic contain an azole ring, such as metronidazole and tinidazole. Azole produce short-lived toxic to the DNA of the sensitive organism. It inhibits the growth of the bacteria. Thank you for watching the video. Please check other videos in the channel and please like and subscribe to this channel.